point out there are still a few seats in the front. Don't be shy about squeezing yourself in. You'll be more comfortable in a seat than you will be standing. But it's great to see the size of the audience here, and it's my great pleasure to introduce our keynote speaker today. So Susan Galbraith is a UK-trained clinical oncologist who did a PhD then in vascular targeting agents and was recruited across to the US by Bristol Myers Squibb. In 2010, AstraZeneca had the very good idea of recruiting Susan back to head up the newly formed Oncology, Innovative Medicines and Early Development Group. And since then, you'll have seen in the media and elsewhere that the massive changes that the oncology portfolio has had in AstraZeneca, the really exciting progress of new agents coming through. I think there's a lot of really exciting things happening in the industry in personalised medicine, and I'm sure you'll all really appreciate hearing Susan's perspective on where that is going and on what you can look forward to in the coming years as both scientists and patients. Susan.
Thank you, Peter. Thank you very much to the organisers for inviting me to come and speak today. Again, I'd encourage those of you that are standing at the back, there are a couple of seats at the front if you want to come and sit down. So the topic I'm going to talk about today is focusing on one aspect of what we're looking at within the AstraZeneca and MedImmune portfolio, which is looking at the discovery and development of drugs that inhibit the genetic drivers of cancer growth. And I'll explain a little bit what I mean by a genetic driver and why I think that's important in a moment. So just a couple of disclosures. I'm delighted to be a full-time employee, very proud to be part of AstraZeneca UK. I hold AstraZeneca shares, which have done very nicely in the last year. Thanks very much. And part of my talk will include a variety of investigational agents that you talk about here, which are not yet approved. So the outline of my talk is basically I'm going to explain a little bit about what I mean about the genetic drivers of cancer and how we can understand them better. Talk about the importance of understanding, monitoring, anticipating resistance that you're going to get when you inhibit genetic drivers with new targeted agents. Talk about the challenges of developing combinations to overcome those resistance mechanisms, and then what is needed in terms of the challenges that presents in terms of the need for innovation in clinical trial design and also diagnostics. And the ambition from all of this is to really change how we're treating cancer. So again, I trained, as Peter said, as a clinical oncologist in the UK, started off at Manchester Medical School just on Oxford Road. And I've treated too many cancer patients for whom you didn't really have the tools in the toolbox that you really would like to have. And I think part of the reason for getting up out of bed every day and coming and doing what we're doing is to try and change that and put tools in the hands of the physicians that are treating cancer patients today so they've got a greater range and they understand better what it is they're trying to do. So as I've said, there are 4 scientific areas of focus that we've got in the AstraZeneca MedImmune portfolio. One very large area is this area that I'm using to bucket together as tumour drivers and resistance mechanisms, and you can see the range of molecules here and their targets listed here. DNA damage repair with the lead compound olaparib is another key area of focus, but we've got other ones behind that, including a very exciting molecule, the WEE1 inhibitor, and others coming into the clinic there. We collaborate with MedImmune in terms of developing antibody-drug conjugates, and there's a very exciting area of immunotherapy with great promise in terms of the potential to change the long-term survival outcomes of patients with cancer. And Robert Wilkinson from MedImmune is going to talk a little bit about that this afternoon in the oncology session. So let me talk again about this genetic drivers, and I'm just going to start by picking on one cancer type, and I've picked lung cancer deliberately because actually it's the biggest cancer killer in the world. About 1.6 million new cases around the world per year. And sadly, the incidence and death rate from lung cancer is going to continue to increase. And you can see this on this graph here. If you're looking at China, the big 5 countries in the European Union and the US, and you can see the projected mortality from this in 2020. And the reason is, sadly, that smoking in many parts of the world is continuing to increase. And in China in particular, there were other environmental exposures which are driving incidence. In addition, we've got an ageing population, and those two things together is what's driving that projected increase in mortality. The other bad news about lung cancer is that the vast majority of people who contract and are diagnosed with lung cancer will ultimately die from it. 86% of patients will die from it. The 5-year survival rate is less than 10%, which is lower than many other major cancers where we've made more significant progress in the last 20 to 30 years. And if you come back home to the UK, 60% of patients in the UK who are diagnosed with lung cancer will die within a year of being diagnosed. And part of that is because we diagnose it late. Only 15% of patients that have their lung cancer diagnosed are still localised to the lung, and those are the people for whom there is some hope of cure. And for over half of patients, it's not just spread beyond the lung tissue to the lymph nodes, but it's also beyond that, and it's already metastatic at first diagnosis. So that's the basic background for those of you that aren't oncologists. I think the other thing to bear in mind is that what we look at and how we describe and categorise lung cancer has changed based on the tools we had available. The original tools we had available was a microscope, and we looked down the microscope and looked at what the cells we saw down the microscope looked like. And lung cancer was divided into small cell lung cancer because the cells were small, and non-small cell lung cancer because the cells weren't small. That was a limited amount of information. Within non-small cell lung cancer, they were then further subdivided into large cell, squamous, and adenocarcinoma, depending on whether they had glandular features for the adenocarcinomas, keratin if they're squamous, and large cell for otherwise undifferentiated. What's happened as we've had the ability to sequence cancers is we've now understood that there are actually molecular aberrations that occur which drive the growth of different segments of lung cancer. And it's actually quite remarkable, I think, that when you think about the thousands of mutations that actually occur in individual cancer cells in patients that have got lung cancer, that the growth of the tumour can be described as being driven by a single mutation in a single gene. But that is the case. And generally, these are mutually exclusive. So KRAS and epidermal growth factor receptor were the ones that were first described. And now what you're seeing is an increase in the knowledge of the segmentation by molecular characteristics of lung cancer. And now you're seeing a definition not based on what the cells look like down the microscope, but based on their molecular characteristics. And what this has made us available to us is a better understanding of truly what the cancer is dependent on for its cell growth. And that's what the term tumour driver, genetic driver, refers to. It means that if you remove that drive from the cancer cell, it stops growing and or dies. And that is, again, as I say, remarkable when you've got the thousands of mutations, but nonetheless true. What's also true, though, is that you will get resistance because those many other mutations will allow for clonal selection of cells that then have an advantage due to a different mutation. So this is the emerging picture of what you saw previously. Large cell adenocarcinoma and squamous, which are subsegments of non-small cell lung cancer. And then what we understand in terms of the representation of molecular segmentation. And I'm going to focus, for the purpose of the next few slides, on epidermal growth factor receptor mutated lung cancer, which represents about 40% of adenocarcinomas in Asian patients and about 15% of adenocarcinomas in Western patients. And what we've discovered is that there are activating or sensitizing mutations in the epidermal growth factor receptor in the exon 9 deletions or substitutions at the 858 position from, as you see here. And those activating mutations decrease the affinity of the receptor for ATP. So here's now a picture of the epidermal growth factor receptor, the 3D structure, which goes through the cell membrane. And here's the tyrosine kinase activating domain of that protein. What happens is when the ligand, epidermal growth factor, binds to the receptor, you actually get dimerization of the receptors, and then you get downstream signaling from this kinase. And you get activation of two different pathways, the PI3 kinase, AKT, TOR pathway, and the RAS, RAF, MEK, ERK, or MAP kinase pathway. And the effect of those is an increase in cell division and a decrease in cell death. And that's what leads to tumour growth. So as I said, this now shows a different picture of the tyrosine kinase domain of the epidermal growth factor receptor. And you can see there's a so-called ATP binding site, or the hinge binding region. And in there you're seeing a drug, ERESSA, which is the first epidermal growth factor receptor inhibitor that was developed by AstraZeneca in its position in that binding domain. What you can also see is that What we're now understanding as a mechanism of resistance to this drug is a mutation in this section. So let me just back up a little bit. What we now know is that in patients that have these activating mutations, about 70% of them will respond to drugs like gefitinib or ERESSA or erlotinib or Tarceva as its trade name. So that's a really high response rate, far higher than we were used to seeing with chemotherapy for either EGFR mutated lung cancer or just wild-type non-small cell lung cancer, where typically the response rates to chemotherapy in first line are in the 20 to 30% rate. So 70% response rate, which means that, just again, back up a little bit for those of you that are not oncologists, a response means that you've seen at least a 50% shrinkage in the bidimensional measurements across a tumour looked on a CT scan. That's what it means. And if you think about that in terms of cell kill, I'll come back to that point later on, that actually isn't a very high log cell kill, but it's enough to detect by, if the imaging folks will forgive me, a relatively crude way of measuring tumour size, which is a CT scan. Okay, so if patients have at least that level of shrinkage, they're called a responder, and 70% of patients will have at least that level of tumour shrinkage, which implies some level of cell kill when they're treated with a drug like ERESSA. However, most of them then relapse about 10 to 12 months after they've had an initial treatment and they've done very well, they relapse. And what we're now starting to understand is what's the molecular pattern that's driving that relapse. And we've discovered that more than 60% of relapses are due to a second mutation that occurs in this position, which replaces threonine gatekeeper with a methionine residue. And what that does, it then means that this protein has a greater affinity for ATP, and it becomes more difficult to inhibit it with an ATP competitive small molecule. So the on switch that was being generated through the sensitizing mutations is back on, and the off that was created by the inhibition of the drug gefitinib or ERESSA is lost, and that's why the tumours regrow. So let me just go into that in a little bit more detail. Here's again another picture of ERESSA bound to EGFR. And what you can see here is the interaction of ERESSA here and the fact that it competes at the ATP for active site, competes with ATP at that active site. So here's ATP and here's ERESSA. So we've known that patients relapse after that length of time with ERESSA for some time now, and there's been a lot of work done preclinically to look at the mechanisms of resistance to drugs like ERESSA or Tarceva. You can see on the right-hand side of this the long list of mechanisms that have been described based on preclinical data for resistance, and you can see there's a whole range of them. Okay. On the left-hand side of this slide are the data that we've actually obtained from patients who've had a subsequent biopsy of their tumours at the time of relapse and had those molecularly characterised. And again, these are the data that show that the prevalence of T790M mutation. And you can see that T790M mutation was indeed seen preclinically, so it does translate through in that sense. But what you don't get from experiments with single cell lines preclinically is a sense of the true prevalence of what's going to happen in the clinic. So you don't get the same sense of the clinical importance, if you like, of the relative resistance mechanism. Okay, I just think that's an important thing to bear in mind as we're trying to understand and anticipate the next mechanism of resistance. And then you see there's a range of other mechanisms, including HER2 amplification, HER2 mutation, MET amplification, transformation to a small cell type, and some combination in a small proportion of patients. And our knowledge about this unknown will continue to evolve over the coming years, I think. So this is, again, a slide that just shows in a bit more detail what happens at a molecular level when you have this threonine to methionine mutation. And you can see ERESSA overlain in that T790M setting in gold. Okay, so you can see here as well that the transformation from threonine to methionine changes the relative position of ERESSA in that binding pocket. So one response to that emerging knowledge was the development of irreversible EGFR inhibitors. And there are drugs, afatinib, which is approved, and dacomitinib, which is still in development, which are irreversible EGFR inhibitors, also based on the anilinoquinazoline structure that you saw for both IRESSA and erlotinib. These compounds contain an acrylamide, and that acrylamide covalently binds to the cysteine residue, which you can see down here. And that results in an irreversible inhibition. And because it's irreversible, you can then overcome the increase in ATP affinity that you've seen. But what you do also see is that these molecules bind also equally well to the wild-type EGFR, the non-mutated EGFR. And that's important because your wild-type epidermal growth factor receptors are present on your skin and the lining of your gut. And so the main side effects that were seen with drugs like IRESSA and Tarceva, and that are seen to an even greater degree with afatinib and dacomitinib, are rash and diarrhea because of that inhibition of wild-type EGFR. And so whilst in theory, preclinically and in vitro, you can overcome the increased binding affinity of T790M with drugs like afatinib and dacomitinib, in the clinic, you can't achieve the exposure that you require to see that inhibition because you're limited by the inhibition of the wild-type EGFR because you're limited by rash and diarrhea. And so the discovery scientists at AstraZeneca laid out a design criteria for the attributes that they wanted to see for a mutant selective inhibitor. And here you see the gefitinib and erlotinib characteristics, very active against the single mutated, the sensitizing mutation. But when you've got the double mutation with the T790M as well, relatively inactive, and they also have activity against the wild type. Here you see the next set of irreversible inhibitors, highly active against both single mutant and double mutant, but also highly active against the wild type. And the profile that was being looked for was activity against the single mutant and the double mutant with a margin to wild type so that you could actually dose up to a level where you could achieve that inhibition of T790M and have activity there. And so what you see here is a plot of the log IC50 of the double mutant EGFR lines versus the wild type. What we're really looking for is something that has increased potency against the double mutant relative to the wild type. And here you see all of the compounds that are in the reversible anilinoquinazoline set, the irreversible versions of these, and a methionine selective group. So that's where the start point came from. And then the chemists then took that start point and evolved mutant selective examples based on the pyrimidine scaffold. And what they saw was that you could get some activity here, but there was a large drop-off between the when you went into cells in terms of potency. So they've then targeted the cysteine using acrylamide in the same way as you've heard previously for afatinib and dacomitinib, and that's improved substantially the drop-off from enzyme to cell in terms of the potency, and it's felt that the position of the acrylamide group was absolutely critical in achieving this. So the candidate drug, now AZD9291, this is its structure, and this is the profile that was seen. You actually got a very potent compound against both the single and double mutant, but with a margin to wild type. Very good pharmaceutical properties in terms of good solubility, good margin to hERG, which predicts for cardiovascular arrhythmic potential, and a low predicted maximum absorbable dose. And this was then tested in vivo. In, first of all, on the left-hand side here, a single mutant model, the PC9 model. And on the right-hand side, the H1975, which has got the double mutation. It's got the L858R substitution and the T790M substitution. So here you see in the exon 19 deleted sensitizing model, gefitinib at a dose that's equivalent to the exposure that you achieve in the clinic. And what you see here, note that in contrast to many tumour growth plots, which are on a linear scale, this is actually a log scale. And you do get some regression initially, but you subsequently get growth in this PC9 model with gefitinib at its clinically used exposure. What you get with a relatively low dose of 9291 is regression and maintenance of regression. You take this into the double mutant model. Again, here you've got 1 mg per kg in red, 5 mg per kg in blue, and 25 mg per kg in green on the 1975 model. And you see that even at a very low dose, you are getting regression, but you get subsequent regrowth. If you then introduce a higher dose of 9291, you subsequently get regression to the point that the tumours are undetectable. And interestingly, at day 200 in this model, the drugs are then stopped and we're not getting regrowth. I would bear in mind, though, that these are engineered cell lines, so they don't represent the heterogeneity and that range of other mutations that you're going to get in a patient with a real cancer. So this is the model binding mode of 9291 in the binding site with the acrylamide binding to the cysteine here. And here's the comparison in terms of, you remember that profile that we were looking for of relative activity versus wild type. And we've used a wild type EGFR model as a substitute, if you like, for measuring the potency against wild type. What you can see is on the right-hand side with gefitinib that you've got the double mutant, very low level of tumour growth inhibition, but good inhibition in the wild type model. With afatinib, you do have potency against the double mutant model, but not as much as you do against the wild type model. And you get, with 9291, a reversal of that pattern with increased tumour growth inhibition in the double mutant model versus the wild type. I'm pleased to say that the preclinical data and the predictions that were made have translated through into the clinic, and these are data that were presented by Pasiani at ASCO earlier this year. With the data that was updated then, there will be an update on this at the ESMO presentation at the end of the month. So what was observed in patients that had T790M mutation is a regression. What this plot shows is, it's called a waterfall plot, and for tumours that are growing, the bars go up. For tumours that are shrinking, the bars go down. So each of these bars represents an individual patient. And what you can see is that for the vast majority of patients, they're getting tumour shrinkage. Again, this is the arbitrary cut point of what you call a partial response. Okay? And the colour code is coding by dose. And the lowest dose that our starting dose was 20 milligrams once daily, which actually corresponds, if I go back, corresponds to the exposure that you see at the 5 mg per kg dose level. Okay? 80 milligrams is the dose that we're choosing to go forward to into pivotal trials, which corresponds to the 25 mg per kg exposure that you see preclinically. But you can see we saw shrinkage at all dose levels, and here's the response rate by dose level. And you can see that at the 80 milligram dose level, we're getting a 68% response in a reasonably large number of patients, and the responses have been highly durable. And again, those will be updated at the ESMO conference in a couple of weeks. But I think, beyond seeing tumour shrinkage, which is clearly exciting, and as an oncologist, whenever you see tumours shrinking in a phase 1 trial, we get excited. But I bear in mind, again, that put that into context, for a tumour to be visible on a CT scan, it just has to have about 10 to the 12 cells in it. For it to become invisible on a CT scan, which we would call a complete response, it can still have 10 to 11 cells in it. Okay, and this is why tumours relapse even when you apparently have got disappearance of disease. So shrinkage is great. That's fine. But the other point of the design of this molecule was designed to see if we could have a margin over wild type. And I think the thing that's most gratifying from my perspective is when you get feedback from patients that are being treated in the trial. There was a patient treated by a great physician, Malcolm Ranson, from the Christie Hospital just up the road, who was one of the first patients to be treated in this trial. And she'd previously progressed after being treated on ERESSA. And her description was, "This is better than ERESSA. It's better than ERESSA because I feel more normal." And she was able to go back and join a gym and feel well. The rates of rash and diarrhea with this drug are indeed substantially lower than have been seen with any of the other EGFR inhibitors. So if you compare with afatinib, which has around a 95% rate of diarrhea, with 9291 at 80 milligram dose level, we're seeing a 20% rate of diarrhea, the vast majority of which is grade 1. Grade 1 is a mild increase in the number of stools per day. Grade 3 diarrhea is requiring rehydration for more than 24 hours. and a rate of still. And with afatinib, you see a 15% rate of grade 3 diarrhea. So it's no exaggeration to say that you've managed to reduce the burden of the long-term toxicity as well as improve the potential efficacy and offer an option to patients who currently don't have one, who've got the T790M. So that's great, but it's not good enough, right? Because we know that resistance is going to emerge to 9291 in the same way that we know with confidence that resistance has emerged to the first generation of EGFR inhibitors. And so what the bioscientists at AstraZeneca have been doing from the early stages of this program is trying to understand and predict what the mechanisms of resistance to 9291 might be so that we can introduce new therapies to help overcome that before we even understand what it is clinically. So they developed resistant cell lines to the PC9 and the double mutant 1975 cell lines by growing them under relatively low concentrations of different EGFR inhibitors. And the resistance lines generated pools of resistant colonies. And these data were presented by Katha Belan at AACR earlier this year. So what has been observed when you do this is that in vitro, the acquired resistance to EGFR inhibitors is associated with increased sensitivity to MAP kinase pathway inhibition, particularly MEK inhibition. And you can see this across afatinib and 9291, and less so with gefitinib. Many of the populations are actually more sensitive then to the MEK inhibitor selumetinib, which is actually also in phase 3 trials for KRAS mutated lung cancer. In a little bit more detail, you can see the molecular mechanism of that inhibition. Again, here you've got gefitinib resistance coming up, and you see T790M as the most common form that's seen across these populations, but you do see novel NRAS mutations also happening. In afatinib resistance, again, you get a mixture of T790M and KRAS gain. In 9291 resistance, we're not seeing T790M come up as a mechanism of resistance. We're seeing NRAS gain or KRAS gain or MAP kinase gain. And for some of them, no genetic change has been detected. So then to follow up on understand this in a little bit more detail, you can understand that if you go back to that signalling pathway that I showed you, NRAS is downstream of EGFR signalling, as shown here. And so when you've got mutations in RAS, you prevent GTP hydrolysis and RAS remains locked in the active state. So you've got that on switch still on, even regardless of what you're doing upstream at the EGFR level. So if you look at this in terms of a Western blot, what you can see is that when you inhibit in a wild-type NRAS PC9 cell line, when you inhibit with 9291, you get inhibition of phospho-EGFR, and you get inhibition of active NRAS, as you would expect. When you've got a novel mutation in NRAS, even though you're getting inhibition of EGFR, you're getting still maintenance of activation of NRAS because the active switch is still on all the time. Okay? And that's true for both the E63K and the G12V mutations that have been seen here. What this graph shows is that these mutations can drive resistance to 9291, and there's a key piece of information that's missing from this slide, as I discovered when I was going through and practicing this, which is that these right-hand two panels, all the columns to the right-hand side of where I'm pointing now, are also exposed to 9291. So let me just walk you through what this means. So over here, again, you've got the PC9 wild-type NRAS model, okay? And if you put in, if you knock out the NRAS signal with siRNA for NRAS, you can see that you're knocking out that activated NRAS signal. But these are wild-type NRAS cell lines, and they grow regardless because you've got the phospho-EGFR in place. In this panel, in all but the left-hand column, you've got 9291 on top, right? So you're getting inhibition of EGFR. But on this panel here, you're still getting activation of ERK, which is downstream, because you've got now the NRAS mutation. If you now knock out the NRAS mutation with specific inhibition with NRAS siRNA, then you could knock out and reduce the ERK signal, and you're seeing the knockdown of NRAS here. And KRAS, in this case, is another control. What that translates through in terms of cell number is the siRNA for the NRAS-specific mutation shows reduction in cell growth, but KRAS siRNA doesn't. And again, that is shown on the right-hand panel, but to a lesser extent. Again, the same kind of experiments done here now with a KRAS gain of copy number, which also shows that you can drive resistance to EGFR inhibitors through this mechanism. So that's what that data does. So that basically just shows us some background molecular understanding of what might be happening as one potential mechanism of resistance to 9291. But what we don't know yet is what the clinical experience and prevalence of resistance mechanisms will be in the clinic. I'll come back to that point later in my talk. So the next question is, if the idea is then that you're going to need combinations to improve on cancer therapy, this is not a new idea. We've been using combinations in cancer therapy for about 50 or 60 years. And the theory has been that always that you come in with things that have got non-cross resistance, and you come in and you work out an acceptable and tolerable schedule of combinations. And if you do that effectively, in some cancers you can increase the cure rate. So all of the cancers that have had substantial improvements in cure rate over the years have done so through the introduction of combination therapies, typically combination chemotherapies in conjunction with radiation therapy. And I think there's no difference actually in terms of how we should approach these molecularly driven tumours. The idea that a monotherapy in a tumour that is multiply mutated is going to lead to an improvement in the ultimate cure rate is probably naive. So I think the combinations are absolutely key to overcome resistance. That gets more complicated. And so when you think about all the things you could do, you could be testing combinations from here to infinity and not make any progress. So you need some approach to prioritising what you are going to do, not just all the things that you could do. And here's my perspective on how to approach prioritising combinations. The first thing is to start where you've got one agent that has anti-tumour activity. And you have to understand that just because you've got the same molecular mutation in a cancer that comes from colon or melanoma, for example, does not mean that it will behave in the same way or respond in the same way to a particular inhibitor. We know that now because we've got effective BRAF inhibitors, and you get very high response rates to those in BRAF mutated melanoma, but you don't get them to a very high rate of response in to monotherapy in BRAF mutated colon cancer. So that's just one example, but I think this general principle of the lineage of where the cancer cell comes from matters as well as the molecular mutations that occur in that cancer cell, and you have to bear that in mind. So start where one agent has anti-tumour activity by understanding the mutation pattern and understanding the lineage and how those interact. Second is you need to understand the mechanisms of pathway reactivation and resistance, and the mechanisms will actually really emerge clinically under clonal selection pressure. And we need to understand that as quickly as possible so that we can devise new mechanisms to overcome that resistance. The third point is that you can dose continuously in vitro for long periods of time, but you might not be able to dose continuously to patients for long periods of time without running into issues. Schedule does matter. Continuous inhibition actually is not necessarily optimal. And that, a common thing that isn't necessarily done well is that we don't often get dynamic data on pharmacodynamics. I know it seems like a strange thing to say, but often you will get one time point subsequently showing that you've got inhibition of a particular pathway. What you actually want to understand is what's the dynamic pattern over time of that change in that biomarker, and what does that inform you about what's happening in terms of signalling within the cell, either for pathway reactivation or emergence of resistance. I think that I'm trained as a clinical oncologist, a radiation oncologist, and we do a lot of radiation biology, understanding cell kill and clonogenic assays. I think there's been a lot of philosophy about inhibition of proliferation in cancer drug development. And inhibition of proliferation is good, but not good enough. And if you actually want to generate resistance, what you do, as I've already shown you, is you use low levels of a drug continuously for a long period of time. If what you want to do is get rid of a population of cells, what you might want to do is actually tip them over into cell death and get rid of them, as opposed to stopping them proliferating. Many of the assays that we use, we need to think about a cell death endpoint rather than just a reduction in cell number endpoint. So when you're trying to kill cancer cells, you also need to think about the interaction between these tumour driver pathways and other mechanisms. So DNA damage response is another area that we've got. Again, that's a good way of killing cancer cells if you've got inhibition of the ability to repair damage. There's interaction between those tumour driver pathways and pro-apoptotic pathways. And of course, there's a whole other area of cell death, immunogenic cell death, and the generation of an immune response in an immunocompetent host, which I think is a key area that we also need to think. And there is going to be key interaction between this, and I think Robert's going to talk a little bit more about that later. And finally, just because you, as I said, just because you can dose a cell in an in vitro dish continuously doesn't necessarily anticipate what the drugs are going to do in terms of interaction in the clinic. We need to understand the tolerability issues and the PK interaction issues, and that can also be used to help prioritise the combinations. So in my view, combinations are key to overcome resistance, and the data I showed you previously that Kath Eberlein had produced was used to help look at the potential to overcome resistance by exposing, first of all, in vitro cell lines to either 9291 or selumetinib and seeing how many cells grow. So these are cells that are seeded and then grown and then divided and counted and seeded again for a long period of time. And what you can see is the increase in cell number over time. There's a gap in the y-axis here because these cells increase dramatically. When you co-dose, this is a double mutant cell line with 9291 on selumetinib, you get a great delay in the emergence of the cell population and the emergence of resistance. And so those data were intriguing and used to help justify an in vivo experiment, which was done in collaboration with William Powell's lab from Vanderbilt, who has genetically engineered double mutant T790M transgenic models in vivo. And what this shows is that in this double mutant model, if you dose with 9291, 5 mg per kg, again, that's a relatively low dose, the 20 mg starting dose in the clinic, if you like, you get some initial regression of the tumour. This is a mouse tumour growing, occupying a large part of the thorax of the mouse, and a regression there. But over a period of time, you get regrowth. And if you then introduce selumetinib, which is AZD6244, you can get further regression. So that combination can restore growth inhibition following progression on 9291. We're now testing that question clinically in a trial that started just last month. So I'm going to change tack now and talk a little bit about a different genetic driver of cancer growth, the PI3 kinase pathway. And just say that the PI3 kinase proteins are not the most commonly mutated proteins in cancer, but this pathway is the most commonly mutated pathway in cancer. And you can see that in terms of either mutations in the PI3 kinase alpha isotype, loss of inhibitory protein P10, or mutation in AKT across different tumour types. And you can see there's a very high prevalence across different tumour types. In addition to mutations, there are other mechanisms that can activate the PI3 kinase activators. So again, some of these growth factor signaling molecules can activate this pathway, as I've shown. There is a drug that's approved, which is an inhibitor of TORC1, which is Afinitor everolimus from Novartis. And that drug has established the clinical linkage of the importance of this pathway in ER-positive or estrogen receptor-positive breast cancer. But there are different isotypes and different mechanisms of activation of the pathway relevant in different patient populations. And I think one of the things we need to do is understand that sensitivity and drive across different patient populations better. And I think another key feature of this pathway that's really important is that you get rapid activation of feedback mechanisms when you inhibit any of these key nodes along this pathway. So we've actually had a large investment across the industry in PI3 kinase inhibitors in cancer. We've had many drugs introduced to the clinic, and none of them to date have shown the levels of response that epidermal growth factor receptor inhibitors have seen in EGFR mutated lung cancer. So why is that? Why haven't we done better? I think there are several reasons. First of all, the first wave of agents that was brought into the clinic tended to be broad, pan, PI3 kinase. The idea was that you might need to inhibit all of the isotypes of this kinase to have activity. But by hitting all of those isotypes, you're typically also introducing significant off-target toxicities as well. So these were relatively poorly tolerated on chronic continuous dosing. Secondly, this key point that you get pathway reactivation. I'll come back to that in a moment. And I think what that does, it limits efficacy by reducing the level of cancer cell kill that you get if you compare it with the amount of cancer cell kill that you get with an EGFR inhibitor and an EGFR mutant cell line. Final point is that some of the mice don't predict well for the clinical toxicity, so rash isn't well predicted from mouse models. And so the preclinical tox profile that you see doesn't necessarily predict well what's seen in the clinic, and the continuous dosing regimens have been poorly tolerated. And finally, in contrast to perhaps the pattern that I showed you in lung cancer, where you've got some mutually exclusive driver mutations, I think there's a higher proportion of codependence of double drivers, if you like, of activity in the patient populations that have been tested with PI3 kinase inhibitors. And so we also need the right combinations perhaps up front, not just as a mechanism of overcoming resistance. So the strategy is you need to understand what that profile is, and our view now is that we need drugs that are designed to be isotype specific and relatively short half-life, so that you can dose them intermittently, and I'll show you some of the data that drives that. And that by dosing intermittently, you allow release of this feedback activation and allow waves of cell kill to occur with each dosing time period. And that also, by the way, improves tolerability. And then finally, you need to aggressively look at combinations and sequences to induce regression. So let's start with the beginning in terms of the patient populations that are most sensitive to different types. What you see here is a PI3 kinase alpha inhibitor that we have that's about to start first in human trials, AZD8835. And when you look at its profile in preclinical cell lines, you can see that the most sensitive cell lines, as measured by IC50, are enriched for cell lines that have a mutation in the PI3 kinase alpha domain. And again, if you then take such a cell line in vivo, this is a MCF7, which is ER positive, and with a PI3 kinase alpha mutation, you can see that it can induce here stasis. Remember, stasis might be good, regression is better. When you actually look at what's happening molecularly in these things, again, over a time course, what you can see is you do get good inhibition of downstream signaling in these cell lines, but that it reactivates even under continuous inhibition. So within hours, 24, 72 hours, you've got pathway reactivation, despite the fact you've got maintained exposure to the drug. And if you look here, if the lights were lower, you could see that there's a brief and transient increase in cleaved part, which is a measure of transient apoptosis seen in this cell line. But it's transient and it stops at this point. And this is a universal phenomenon with agents that inhibit this pathway. Again, you can see it here in terms of what's actually happening downstream. FOXO is a gene that's under the control of PI3 kinase, and you can see nuclear translocation. But if you look at the timing after dosing, again, this is peaked at 2 hours and then returning back at a later time point. So how to overcome the pathway reactivation? What you can do is add in another component of the pathway to overcome this feedback inhibition. And this looks at now a cleaved caspase 3 endpoint on immunohistochemistry. And you can see the brown cells have got cleaved caspase 3 and they're going through apoptosis. And if you dose in combination with AKT inhibitor AZD5363, you can get increased apoptosis. And you can get that most notably on the right-hand side if you dose concomitantly, but you can also get it if you come in sequentially. So you're basically generating the feedback activation, then you're coming in with a different molecule. What you can't see from this experiment is actually the concomitant dosing isn't as well tolerated as the sequential dosing. And again, that comes back to what you're actually trying to do is get a better therapeutic index, not just an increased cell kill. Final point I want to make is now with a PI3 kinase beta inhibitor starting off is that, again, this point I was making about the lineage matters. It's not just that you get pathway reactivation within the PI3 kinase pathway, but if you suppress PI3 kinase activity in a cell that is from the that's driven by the androgen receptor, you will get upregulation of AR-related genes, AR and AR-related genes PSA and TMPRSS2. And it's a bit like a seesaw. You inhibit PI3 kinase, you get upregulation of the androgen receptor-related genes. If you inhibit the androgen receptor-related genes, you get increased activation of the PI3 kinase pathway. So if you really want to kill cells, you don't just have to inhibit the PI3 kinase pathway hard, but you also have to inhibit the androgen receptor signaling pathway. And again, you can see this in terms of maintenance of inhibition of tumour growth through the combination of the two pathways. And this work was done in collaboration with a group at the Erasmus University. So these are data that come from another collaboration we have with Neil Rosen at Memorial Sloan Kettering. And again, this shows about the combination of an alpha and a beta PI3 kinase inhibitor. Again, this is in a AR-driven background. So AR-driven background with PTEN loss, it's a beta PI3 kinase driven process. And if you inhibit with a beta inhibitor, you get inhibition transiently of the beta pathway. But it comes back. If you inhibit with an alpha inhibitor, nothing very much happens because this is a beta-driven cell line. If you inhibit with both, you get much more sustained inhibition of PI3 kinase signaling, and that translates through to a much greater rate of cell death. And if you look at that in vivo, what you can see here is tumour growth bars very similar to the tumour waterfall plot I showed you for 9291. In blue is the beta inhibitor alone, which doesn't do a whole lot. In red is the combination of a beta and an alpha, so you're getting very good PI3 kinase pathway inhibition, more sustained in this case, but again, not a huge amount of cell kill here. Variable, but not a huge amount. If you dose the beta inhibitor with an AR antagonist, which is enzalutamide or medivation 3100, you get an increased level of cell kill. And if you put the three drugs together, you're really getting now a much more impressive level of cell kill. So combinations are critically important in the context of the lineage to cause regression. I do believe that PI3 kinase pathway inhibition needs to be combined with the relevant inhibition of the lineage from another component. It doesn't have to be AR in an AR-driven cancer. It can be ER in an ER-driven cancer, or it could be chemotherapy. There are different ways of achieving the same goal. The other point that I would come back to is, again, you've got to actually come up, if you're going to come with combinations, to tolerable regimens. And what we've shown here, this is work that's been done by Kevin Hudson and other people in the bioscience group, in an ER-positive, HER2-positive PI3 kinase mutant cell line, is looking at continuous dosing, which is the red line, 25 mg per kg, twice a day continuous. 2 days on, 5 days off, in blue. And you can see in a tumour that grows on the side of a mouse, you have a relatively high proliferative index. And if you stop dosing for a period of time, you do get spikes of regrowth. If you dose Monday, Thursday as opposed to Monday, Tuesday, you can also get stasis. But what you can then do with the Monday, Thursday regimen is achieve a higher dose which is tolerated and a greater level of tumour cell kill and therefore regression. Now you can just see from what I've shown you that actually there's a huge amount of work involved in this, working this out preclinically and then thinking about how you translate this to the clinic. This is not simple to do, but if you wanted simple, you should have done rocket science because physics is easy and biology is complicated. Okay. So does this translate through to the clinic? Intermittent dosing has been tested now with our AKT inhibitor in the phase 1 clinical trials, and the data that we've shown actually supports intermittent dosing. So again, just bear with you. We're using here hyperglycemia as a pharmacodynamic marker for inhibition of the PI3 kinase alpha component of the pathway. We know that if you inhibit that, you will get a hyperglycemia. So you can see the rates of hyperglycemia that you see at 400 milligrams, 480 milligrams, and 600 milligrams on a continuous dosing regimen on the left-hand side. And then 360 milligrams, 4 days on, 3 days off, 480 milligrams, 4 days on, 3 days off, and 600 milligrams, 4 days on, 3 days off. And again, you can see that you can actually get to and tolerate a higher dose intermittently, which produces a higher rate of hyperglycemia. But interestingly, the things that are less well predicted from the preclinical models as dose-limiting toxicities of diarrhea and rash, but are a real problem in the clinic, you're getting lower amounts of those on the intermittent dosing. So the next question you're going to ask me, of course, is, well, does it work, though? Does it actually shrink tumours? And I haven't got the AKT data to show you for interest of time, but the answer is we are seeing responses in patients on the intermittent dosing schedules with AKT inhibitor. There's data from Uday Banerjee presented with our dual TORC inhibitor at ASCO this year from the Institute of Cancer Research. Again, in combination with paclitaxel here on an intermittent dosing schedule with 2014 dosed 3 days each week. And what he saw is a high partial response rate in this phase 1 trial in a patient population that's highly refractory. 0% grade 3 toxicities at 50 milligrams BID and a high disease control rate, which is very encouraging. We've got 2 out of 2 partial responses. So when you've got 100% response rate in small numbers, you also know that it's only going to go in one direction. But nonetheless, even those small numbers, encouraging in these patients because they'd both progressed on prior taxane therapy. So I think this is very encouraging data that suggests that the preclinical data saying that we can get tumour regressions with intermittent dosing is translating at least into the phase 1 trials, and of course we're going to be testing that more robustly in phase 2 trials shortly. I would also say that there's other potential ways that you can have interaction of the PI3 kinase inhibitors with the DNA damage response, and I'll just show you very briefly that you can see this in this plot, again in an ER-positive, HER2-positive patient tumour in vivo model, where you can get enhanced activity by dosing them continuously. And you can actually see downregulation of homologous recombination genes by the addition of 214, which may drive the sensitivity to olaparib, which is works particularly well in cells or in patients that have got a homologous repair deficiency. I'm going to skip over that for reasons of time and just say briefly now that, okay, so you take with me so far the conclusions that we've got about that we need to understand the molecular drivers, and that we need to understand and anticipate the mechanisms of resistance, and we need to develop combinations. All of those have implications for clinical trial designs. And I would say that the current paradigm for clinical trials isn't working very well for patients, for physicians, or for pharmaceutical companies. So when you think about a low-frequency event, a 2% mutation in lung cancer segment, for example, that you have to screen a lot of patients to find that 2%, and that costs a lot of money. There are issues in terms of the sample quality. There are issues in terms of the calling of whether a patient has a mutated cancer or not. There's therefore difficulties that come in terms of cycles of repeat of standard diagnostics and whether you've got consistency between different calls. You've got the current regulatory requirements to validate the companion diagnostics. And for all of these reasons, we need to change how we're doing it. I'll give you a very real example. This is only partially anonymised, but this is a real example that we've had. Right, so the first thing that might shock you is how much money it costs to treat an individual patient in a phase 1 clinical trial. That's the real cost. Okay, and then if you're looking at a marker at 2% rate, okay, at around $1,000 an assay, and a 15% test fail rate because you haven't got the right amount of tumour or something else happens, that this is a screening model, so you're going to discard an awful lot of patients for the patients that you put on. So for a 20-patient study, that translates through to nearly $2 million, with the size of a screening effort that's more compatible with a phase 3 trial rather than looking for an effect size in a phase 1 population. The physician's experience is it takes a lot of time and effort to get a clinical trial up and going and approved at their site, only to have a large number of patients that they screen through they're not able to be put onto the clinical trial. Very few go on, so they get very little experience with the drug. And from the patient's perspective, they just want to be able to get a drug that works, right? I mean, it's just really clear. They really care whether they've got an FGFR amplification in their tumour, but they do want to get on a clinical trial. They have to go through this long consent form where you explain the potential for this exciting new drug and the benefits and the side effects that it may have. They get to the point where they want to sign it. They sign on, they get the test, and then they're told they can't have it. You can imagine that in the setting of a patient with refractory cancer, where the emotional burden is already high, that's not an easy experience for patients. So there's got to be a better way, and in fact, I think there is a better way. And one example of that is so-called basket trials. One example here is the Lung Master Protocol, which is being run in collaboration with Friends of Cancer Research, in collaboration with the FDA, the FNIH, the NCI, SWOG and ECOG. What they're doing is basically having multiple diagnostic pathway and then putting patients onto one of 4 different drugs coming from multiple different pharmaceutical companies. So our FGFR inhibitor, AZD4547, and our anti-PD-L1 antibody that Robert's going to tell you more about, MEDI4736, or Genentech compound, or an Amgen compound, or a Pfizer compound, depending on the molecule that the molecular diagnostic that they've got. This means that the screening effort doesn't just go to one drug, it can go to many different drugs. The patients are more interested because they've got multiple chances of getting onto the right drug. The investigators are more interested. From my perspective as well, it also means it's a much more efficient way of getting the answer to the question, does the drug work? Another example here in the UK is a collaboration that we have called the Matrix National Lung Trial that Cancer Research UK is running. Again, same principle, phase 2A, signal-seeking arm with monotherapy. And again, we're using next-gen sequencing for screening. And the screening is delivered via 3 technical hubs, so you've got some degree of standardization. And there's around 12 of the AstraZeneca compounds and 2 compounds from Pfizer that are participating in this trial. I think these are just some examples. There are multiple other things that are happening across different tumour types. This is a national effort. It requires a national effort and a national infrastructure if this is going to work. But I do think we need to change how we're doing things, and that means we have to have this infrastructure in place for it to work. What this means, if you look at the clinical trial, is you've got now a range of different mutations that can occur that will lead you down the pathway to different drugs, again, across different things, both in adenocarcinoma and squamous cell carcinoma. This might look more complicated, but as I said before, biology is complicated, cancer is complicated, and actually, if we want to make a difference to the disease, we have to accept the complexity, not stick our heads in the sand. Also means that we've got to have in place the right diagnostic tools. Next-gen sequencing is very exciting. It's rapidly evolving. It's moving rapidly from research to clinical grade. Illumina is working with the FDA for clearance for MySeq. LifeTech's expecting FDA clearance this year. There's companies like Foundation Medicine providing expert development and interpretation. And there's anticipated that we will get approval of a next-gen sequencing companion diagnostic test in the near future, and we hope to be part of it. But let's understand that besides all of the hype and the hope, there are challenges associated with this, right? Different platforms yield different interpretation. Calling it right is difficult, particularly for large genes like BRCA and P53. Many of the tumour suppressor genes have large numbers, thousands of mutations, much more difficult than hotspot mutations, for example, in EGFR. Sample quality is really important, and inter-platform and inter-laboratory concordance is critically important. And so far, despite the hope, we haven't yet got a companion diagnostic approved by the FDA that's based on an NGS platform, but I'm hopeful that we will do soon. And I think one of the point is that I've said before, we want to monitor what's happening in terms of tumour cell kill, and that the imaging tools, whilst helpful, aren't sensitive enough to really tell us that you've got the level of log cell kill that we really want. I think circulating tumour DNA, so measuring the mutations that you can detect in tumours in the peripheral blood, the sensitivity and specificity of these techniques is now improved to a point that it's potentially practice changing. And we have submitted an application for ERESSA to be approved on the basis of a circulating tumour DNA test in the European Union, based on these data, which show that the response rate in response to ERESSA, if you're just detecting a circulating DNA test for EGFR mutation, is as high or higher than it is if you're using that test based on tumour tissue. And the advantage, of course, for the circulating test is you can get serial changes over time, and you can watch what's happening to the mutational load, and you can do that now with 2 logs, maybe 3 logs of sensitivity in change. So you're starting to be able to quantitate much better the change in cell kill and watch resistance as it emerges. So we're going to be using this also on the trials for 9291. We already have data, and I think as its utility as a means for potentially detecting resistance as it emerges is also showing promise. So come back to my last slide now, which is basically come back to that point. I do think we're actually in the golden age of cancer drug discovery, really. I think we've got great potential to mine the data that's emerged to understand the genetic drivers of cancer, to understand, monitor and anticipate the resistance, to develop combinations. But we have to change almost everything that we're doing if we're going to do that effectively. But the goal is one that's both ambitious, but I believe achievable. It's not beyond the wit of man or even women to help redefine the cancer treatment paradigm. And I'll end by just saying cancer development, drug development is a team sport. For those of you in the UK and Europe, it was the end of the football transfer window yesterday. I happen to support Manchester United, and I was very interested in the cost of the transfer. It would be very interesting if our scientists were paid as much as the footballers. These are some of the stars from the AstraZeneca team that I think deserve acknowledgement, although I don't think I'm paying any of them the same salary as Di Maria. But anyway, I'd just like to point out many of these people contributed. The chemists, Mike Waring, Richard Ward, and Ray Finlay were the discoverers of 9291. Darren Cross is the lead bioscientist. Kath Eberlein I've quoted as doing a lot of the work. Barry Davis, Simon Barry, Kevin Hudson, Stephen Green worked on the PI3 kinase pathway agents. Mark O'Connor is on the DNA damage response agents. Graham Smith and Paul Smith, key parts of the bioscience group here. And of course our external collaborators I've talked about at Memorial, MD Anderson, Vanderbilt, Erasmus, CRUK and Friends of Cancer Research. And with that I'll stop. Thank you.
Any questions for Susan? We've got a question. Front row there, Alan.
Susan, as an alternative means of overcoming the resistance to growth factor inhibitors, can you comment on the utility of the current crop of checkpoint inhibitor immunotherapies that are coming through?